His topic is biological response to stream restoration. Mark, welcome. Thank you for joining us and volunteering to participate, or strong-armed, whatever the case may have been, but we're happy to have you here.
Great, thank you. So should I share my screen now? Just go ahead.
Yes, please do.
Okay. Do you see the screen?
We sure do.
Okay. Well, again, I want to thank everyone for the invitation. Really always appreciate the opportunity to talk about what I like most, which is biology, and in this case, biological response to stream restoration. And well, first of all, I want to provide a little bit of context. So this talk was by invitation originally from the Chesapeake Bay Program Scientific and Technical Advisory Committee, the STAC, for a workshop that they did just a couple of weeks ago on stream restoration. And what they asked me to do is to put together a synthesis talk on biological response. So it's largely some of the stuff that I've done, but also other colleagues of mine and other literature that I pulled together. So first of all, I really want to thank all those other researchers that took the time to talk to me about this and provide me with some examples. So thanks to them, but also, of course, need to say that all of the conclusions are just mine and no one else's. Sorry. Okay, so because it was a synthesis talk, I started with the end. So you guys can look at this and decide whether you want to continue to watch it or not. But this is what I'm calling What's Up with Uplift. Because biological uplift is a big topic a lot of people are interested in. And so the first conclusion is that biological uplift is rare, you know, in the literature and the studies that I've looked at, that the limiting factors affecting biological uplift are many and they're often elusive. Therefore, designs of stream restoration should address those limiting factors. Ultimately, the watersheds that they are in really determines the potential for uplift. And therefore, the threshold for intervention for doing a stream restoration should be relatively high and based on certain criteria, which I will get to in the talk. So starting with the basics, you know, we imagine, you know, again, streams are dynamic systems. So if you think of a degraded system with some variability on the bottom series of points, that's the green, just changing a little bit in stream quality over time, however you measure it, whether you measure it through diversity of species, indices of biotic integrity, or whatever. And then you have some reference, which is a higher quality stream that you would like your stream to attain. And that would be the blue here along the top, again, with some variability. And of course, your restoration starts in the red on the left and just begins slowly and with some variability to get to approach that higher quality. So that's what you're looking for. You're not looking for a single jump, you're looking for some change over time. So really, again, as I said, this is a synthesis talk. So I went to some literature, and again, I talked to a number of investigators. And so I just have a couple of examples here to start off with, just looking at invertebrates in particular. So I'll focus mostly on benthic macroinvertebrates and fish, but I'll mention a few other things as well. So Margaret Palmer and her colleagues did a big review paper in 2010. And of the 78 stream or river restoration projects that they studied, there were statistical increases in invertebrate taxa richness in only 2 of those cases. So again, it's a rare occurrence, even though most of these projects enhance physical habitat heterogeneity, which is often a goal of stream restoration. So she suggested that, quote, managers should critically diagnose the stressors impacting an impaired stream and invest resources first in repairing those problems most likely to limit restoration. So that's the theme that you'll hear throughout the talk. And then just turning to fish, again, and this shows another review paper, this one by Phil Roney and his colleagues in 2008. And they looked at actually 345 studies and again found that they rarely demonstrated uplift. But in a lot of cases, they attributed that to the fact that the monitoring, the research was relatively in short duration and fairly limited in scope. But they did notice that there was some improvements in in-stream habitat that increased local fish abundance by reconnecting habitats, doing some floodplain rehabilitation, which is common. But the failure in general was attributable to, again, to not assessing the historic conditions and factors limiting biota. Again, that's the main theme here. So we really need to understand water scale processes and and monitor at the appropriate scales. So after those two reviews, again, there's a lot of information in there, and I'm just going to jump to the work that's done in the Chesapeake. Again, a couple of my colleagues, Scott Stranko and Bob Hildebrand, have some nice papers out. And these are the two studies that I did with my colleagues here at the bottom, and I'll talk to each of these in order here very briefly, obviously. So the first one is just talking about Scott Stranko's work. Scott is the director of the Maryland Biological Stream Survey, which I've worked on too since 1993. They have done more than 5,000 stream sampling sites and have taken those techniques to stream restoration sites as well. So they have a large suite of reference sites of various kinds in all physiographic regions and stream sizes across the state. And what you see here is a simple cluster analysis where if you're looking at the solid triangles, those are the urban restored sites, and you see that they are clustering with the other urban non-restored sites rather than with reference sites, the solid circles, or any other non-urban sites. So again, you're not seeing much movement from the urban sites that are not degraded, or that are degraded when they're restored. They're really not approaching the reference sites that you might hope to. And taking a similar cluster approach is another study done by Bob Hildebrand at the University of Maryland Center for Environmental Science. And he took a bunch of sites in central Maryland in the Piedmont and looked at, again, restoration sites that and biological changes after restoration. And you can see he had a couple of different kinds of references. He had these upstream and downstream references in addition to his restored reach. And those are all clustered together there in the middle with the red, blue, and black. And that the reference sites comparable to what Scott Stranko had are the greens here. And again, they cluster quite differently, the reference sites. So we're not really seeing, again, much movement toward the reference sites from any of the restored sites in the studies that he looked at. He looked at about 40 individual sites. Digging in a little bit into Bob's work, again, the focus of restoration is often physical habitat improvement, and that did occur. If you look here, you can see that his restoration sites, the blue in the middle, were as good or better than any of the references they had nearby. So that was achieved. Physical habitat was improved, but again, not the IBI or the benthic. We should looking at benthic macroinvertebrates. Here's a couple of other graphs from Bob's work. You can see on the left that he's showing the benthic IBI on the vertical axis for, again, the three different kinds of sites, the actual upstream and downstream references in the red and black, and you see those track almost exactly the same according to percent of impervious surface, but that the restored sites actually do a little worse. They do a little worse at higher impervious areas. So again, not improvements that you might hope to see there. But a little hint of optimism is looking at the right graph. You'll see that the IBI scores over time didn't change significantly, but there is a suggestion that maybe there was some improvement once you get out to that, 10-year plus period. So that's something to keep in mind as well. So now I'm going to jump into the two studies that I authored with my colleagues, in this case with my Tetra Tech colleagues, but also colleagues at the University of Maryland Chesapeake Biological Laboratory here, Ryan Woodland. Did a lot of work with me. And the topic here was vertebrate communities. So we're focusing on fish and reptiles and amphibians and what are called regenerative stream conveyances. I don't know if that topic is familiar to, it's certainly becoming more and more popular here in the mid-Atlantic, specifically in the coastal plain region. But what you see here is our conceptual model. We thought that the idea here was that perhaps we were comparing our restoration response to the wrong reference, and that we really shouldn't be comparing it according to that blue arrow, which is saying that a restored stream, an RSC, to another single channel high quality stream, but we should be comparing it to a high quality stream wetland complex, the green arrow here. So that was our presumption that perhaps we have a better trajectory toward that reference site. And so we did this study looking at, again, a bunch of sites, dozens of sites in the central part of Maryland in the coastal plain. And you can see here, this is the way all the graphs will be set up. On the left, you have those single channel streams that are high quality. These are all drawn from the MBSS study that I mentioned, the 5,000 streams in Maryland. That's the blue. The green is the stream wetland complex, again, high quality. And then the low quality or degraded streams are in the gray, and then the RSCs are in the red. And so just bottom line answer, you look here at the fish IBI, which is a measure of diversity and abundance in fish. And it shows that again, RSCs are definitely not significantly worse. They are significantly lower than either of the reference sites, even though the one we presume they might approach. And they're not significantly different from the degraded sites, the presumed pre-construction condition, although there's a hint here, again, that they're a little bit better, but not statistically different. So breaking that down a little bit, looking at the fish diversity, richness, area richness, and Shannon Weiner, you see that again, that's why you saw the previous graph. Again, the reference sites are better in all three diversity measures than the RSCs, again, which track with the degraded sites. Interestingly, however, the herpetofauna, and again, that's reptiles and amphibians, and in this case, it's mostly frogs. Again, we're in the coastal plain. Salamanders don't play a big role, and reptiles are, again, relatively rare. But you're seeing here not much of a difference. It's sort of similar, maybe a little worse, but generally not much different than the reference sites. And the reason for that is that there was some difference with frog abundance. Quite a few frogs, and again, if these are stream wetland complexes, it makes sense to have more frogs. And you can see that there actually is some differences among the frogs, and that's what the amphibian numbers on the left show you. And breaking this down into sort of a summary table here, you can see again, we're talking fish and frogs compared to these various different references. So again, a red bar is the RSCs. This is our test sites, the restorations, and the two references, the blue and the green. And you can see that, again, the richness is not improved, but abundance actually is among the fish and among the frogs as well. So let's talk more generally about biological uplift and what I've just described and what it really means. So I want to first of all say that as Bill Roney mentioned in his stuff, there's just lots of studies where it's hard for him to figure out what the response was because it wasn't long enough, it wasn't capturing the right habitats, it wasn't doing the right measurements, et cetera. And that's true. It's just a lot of people are getting projects in the ground. It's important to do this for a lot of different reasons. It's an important tool in the toolbox for a variety of reasons, but only a very small proportion of projects are monitored. So we really don't know what's going on there. And when they are monitored, there are limitations. Most are monitored after construction. So again, we don't have that pre-construction, which is really would be important because then you know exactly what the pre-condition was. As it is, we have to do some inference and we have to look at reference sites that are different, either upstream, downstream, like Bob Hildebrand did, or just references like I did or Stott Sranko did. And those references that we're using may be less degraded than the site was, pre-construction. So that's maybe not, they will be less degraded, but they may not be appropriate for that site. They may not have the same history. It's an important thing. We don't know exactly what the previous history was at that site, because again, if we're using a different reference than the pre-construction reference, and there's more variability when you do that. Obviously, if you're going to different areas of any kind, different stream sizes, land use, landscape conditions, there's going to be more variability. That can mask a signal. But again, I think the results I showed are pretty conclusive. But again, if we had more data and we had more appropriate references, we'd probably feel better about it. The best way to do that, of course, is the before-after control impact design. But that, again, requires that you have before and after construction at both the reference site and the restoration site. Those are even rarer, probably, than the instances of positive biological uplift. But there are positive examples. So let me jump to what we have here. Some examples. Again, it's very kind of these folks to talk to me. These are people I've met over the years. So Dave Penrose down in North Carolina did work with invertebrates at 3 sites that I mentioned. Bob Siegfried has worked on some fish down in Virginia. And Amy's got an interesting paper in Kentucky. So here, jumping to one of Dave Penrose's papers, this is Foster's Creek. And here he showed that, different stations, if you look at the different colored lines, those are times, over from 2018 to 2022. And you see that the highest line is the later line, and you have an increase in EPT, which again are tolerant or intolerant to water quality invertebrates, basically mayflies, stoneflies, and caddisflies. So another example, again, digging into the benthic macroinvertebrates that Dave did. This is Dotson Branch, and again, you're now looking at I think over a 4-year period. You can see each of those changes, and basically with a little variability begin to get higher and higher levels of EPT and other biotic measures. So basically the intolerant taxa were increasing in his restoration site here. This is the case where he added riparian vegetation. So again, we're beginning to see, well, this is a why, perhaps. Maybe that's why he had success there. Here's where he added a riparian wetland at Carolina Bison site. And again, we have 2 sites here on the restoration, 3 years apart, and the results, and you can see again a significant increase in taxa richness and EPT abundance. Moving on to fish. So here we've got the work from Bob Siegfried in Virginia. And again, what you've got is a long list of species that were there at pre-construction and then post-construction. So this is, again, some valuable data. And a couple of years later, 2 years later, and then 3 years later, and you're seeing some additional species show up. Not a lot, but some different species. This is the case where he removed a blockage. So again, that makes sense. If that's the limiting factor that prevents the migration and movement of fish, and you can remove that, you should see a biological uplift. Here's a case where he increased riffle habitat, Little Westham Creek. And again, you can see various minnow species that showed up that weren't there before. Again, 2, 3, and 4 years after construction. In this case, they increased the riffle habitat to about 50% of the channel. Here's another interesting case where, again, this is called Proctor's Creek. And the idea here was that there are actually some acidic soils in the site that they believed were limiting the fish community. And once they covered those in the restoration, they did get, again, a number of species returning to the site. So again, this is another case of addressing a limiting factor that's very specific and seeing a positive biological response. And the last example here that I'll mention is the one in Kentucky, where they found out that the EPT biomass was 3 to 4 times greater in these pools that were created. So one of the take homes here was, again, you need to look across the different habitats. And if your design is creating different habitats, you need to make sure you measure those properly and not just do maybe a standard monitoring program so that you can make the proper comparisons. And again, here's actually the last one on the biological uplift examples. Talking about Hyporheic taxa. So here's something that most people don't think much about. Again, these are the organisms that live down in the sediment zone that interacts with the stream channel. And in this case, we're talking about the Robertson paper. The benthos, the benthic macroinvertebrates that I've mentioned, did not improve after restoration, but the Hyporheic taxa did increase. So another part of the ecosystem to look at to determine biological response. Here's the actual data. So the red ones, the red lines are the Hyporheic taxa, which you can see have increased over time. So, okay, so that's the data. That's the story that I got from, the papers that I looked at and the people I talked to and the work I did. And so really what's going on and what's the why here at this point? We sort of saw what the what is. So what is the why? And so these are the things that I think are the main limiting factors that have been identified. Habitat, which often is improved, flow, which can be improved or not, water quality, typically not. But then there's also time and proximity to sources. These are all factors affecting biological response. So now I'm going to jump in again and just mention some examples from the studies that I did with my colleagues. Again, here's the RSC paper. We showed that habitat was not limiting, that these RSCs did create more habitat in their streams. Basically what these RSCs do, they're relatively small watersheds, and typically stream wetland complexes are in larger watersheds, often beaver influenced situations in a low relief landscape, but they pretty much successfully reconstructed those habitat types on a smaller scale. And you can see from the next graph that if you look at all the different measures of habitat features, they were all comparable to the references, with the exception that there was less buffer because that's the construction removes some riparian vegetation, and there were more cobble based on things that were added to the stream. Otherwise, the habitat was very similar. Flow and water quality remain limiting, however. I mean, this is the kind of stuff that when you go in and you do a physical restoration, you may not affect these factors significantly, and those can still limit both vertebrate and invertebrate uplift. This is what we showed in this paper. Again, we're focusing on vertebrates here. It did not attain the reference dissolved oxygen or conductivity, and the flow levels may or may not be obtained depending on the site. And another example I got from Chris Ruck at Fairfax County, where again they did some similar restorations, but again, because they had higher temperatures from less riparian canopy, they caused a trophic cascade of algae that ended up reducing their minnow community. Here are the actual bar charts, again showing the same reference on the left, blue and green, and the RSCs in red. And you can see that in each case, worse water, worse dissolved oxygen, higher temperatures, and higher conductivity, which can all be problematic for the species we're looking at. Again, the vertebrates, specifically fish. You can see that the fish don't like the low dissolved oxygen. Temperature is not as big a factor in these particular sites, but conductivity was definitely a negative factor with them. But interesting, looking broadly again at the ecosystem and the various biota, the herpetofauna, again, principally frogs in the study, were not reduced by water quality. Again, amphibians have a double life and therefore are not as tied to water and therefore its quality. And then we jump to time, which I think is another thing that we need to keep in mind. The research looking at all these different studies, we saw in my study, something that matched what I saw in the literature. I remember first reading through the literature of Margaret Palmer saying, if you're seeing anything positive, it looks like it's 8 years out, and it was about 7 years in this study where we saw some suggestions of improvements. Again, not significant, but suggestions. The fish abundance increased, but not the diversity over time. Herp abundance and diversity did increase over time. Again, the number of frogs increased about 8 years out. So again, that sort of magic number. Don't obviously want you to read any data here, but you can see from the colors, all of the pink is pre-construction. The green is when the site was constructed. These are the actual sites we used in our study. And then the blue are post-construction. So you can see there's quite a bit of variability. We didn't use anything. There wasn't at least 2 years post-construction because we know that 1 year is still recovering. But it really more time. You see the ones that have a longer number of years post-construction, they were the ones with the highest response. So time does seem to be a significant factor. You can see here the things that, again, the fish abundance did increase. I showed that and mentioned that before, but not the diversity over time. You're breaking this into different time blocks, and the herps increased both the abundance and the diversity over time. And here's sort of a better graphic, just to again sort of show you what was going on principally with frogs, that they sort of were going along at a certain level for a few years, and then they got to be at maybe 6 years, and they started going up, and then after 8 years, it sort of plateaued out. So again, an idea that time can be an important factor in studying biological response. Movement barriers is another one. Okay, so now I'm getting to talk about time, now we're talking about space. And the proximity of source populations is important because animals need to move, and whether they drift as invertebrates, have aerodispersal as invertebrates, or travel as fish. Headwaters and other streams may lack upstream populations, so you may not have any source really at all to come down. If you have physical barriers, they obviously limit fish movement, whether those are culverts or whatever. Water quality can be a barrier too. If it's really bad water quality, they may not transverse it, even though they have the physical freedom to do so. So we should keep in mind that when you're looking at restorations, the poor dispersers will take longer, if ever, to repopulate your site. So this is the actual study that I did with Chris Swan at the University of Maryland, Baltimore County. And what we just did was a meta-analysis of a bunch of restoration sites, again, in Central Maryland. And again, another big chart showing the various site factors, but the distance, the one highlighted in black, shows a significant effect of distance. And what that means is that the biological quality of a site was higher the closer it was to other high quality sites. So if a site is sort of out there in a biological desert without high quality sites nearby, it's less likely to have a positive biological response. So that's the what and the why. And then, so, you know, how do we make this better? And so what about design? So what do we do? And I want to let you know these figures were used with permission from an artist in Finland, but I think they're pretty cute. So let's think about how we should do this. So if you want to restore a stream, you should look at sites that have few or, just one limiting factor or a few that you feel like you can address. Because if you can't address the stressors, you're not going to have biological uplift. Now, you may do restoration for lots of other reasons, but if you're doing it for uplift, you really need to think about stressors. Examples include looking at point sources like acid mine drainage. Scott Stranko has a great example in the Youghiogheny River in Western Maryland, where it was basically wiped out by a major acid mine spill 100 years ago, and now it's coming back because that's been addressed. Agricultural settings are also good examples for restoration because typically there's no riparian vegetation. If you can increase that, the in-stream wood as well, and reduce temperatures, you can definitely get an uplift on particularly, trout in the Upper Beaver Creek as an example. And then think about really low quality habitats that could be made better. Things like upstream gullies with little or no water or habitat. Those kind of streams, if you can effectively reduce the sediment and erosion, then you can, obviously get water back. And if you get water back, you can get biota back. Upstream habitats, again, which is the focus of a lot of restorations, sometimes can create uplift. Again, if you don't have other overriding stressors, you can increase vegetation, roots, wood, stability of the substrate. All those things are good for biota. But if you're doing that in a water quality compromised situation, a landscape, really what you're going to get are the water quality tolerant species, and the sunfish and the frogs would be examples of that. Hydrology can sometimes create uplift. Here's Margaret Palmer talking about in another paper where evidence suggests that restoring particular facets of flow regime can produce desirable conservation outcomes, but context is paramount. Again, flow depends on what's the habitat requirements of the species. And just going beyond discrete flow events and really looking at subsurface flow and everything else out there is important to dealing with whether flow is going to cause biological uplift. And this is Phil Roney. Actually, I sort of like this. This is a four-tiered way of looking at restoration, again, focusing on water quality solutions. So he says, protect your high quality habitats, which I totally agree with. Then look at water quality and quantity. Deal with that as a stressor. If you can eliminate that limiting factor, then you can restore those watershed processes that we typically do. Connect habitats, reduce sediment, improve hydrology, and connect to the floodplain. And once you've done that, then you can add habitats that can increase biological diversity. So what does that mean? So the bottom line is that the watersheds really determine the potential of uplift. If you're trying to improve the biological community, you need to be aware that particularly all watersheds in the United States are modified from historical conditions. So they're changed. So they have some limits. The ones that are best remaining may be islands that are limited by space and ability to recolonize. Impervious surfaces really are a major uplift because they create these flow and water quality problems, and that occurs even in stream wetland complexes, which we sort of were hoping was not the case, but was shown to be the case. Watersheds often pose uncontrollable and maybe even unknown stressors. What Bob Siegfried has said, I sort of like the way he puts this. He says, if you're thinking about what your potential could be, what your goals should be, what is already there, what can live there in that watershed based on limiting factors, and can it get there? Thinking about that. So really, the goals of a stream restoration should be based on these expected potentials and whatever observed you get relative to that, and trying to maximize that observed over expected. And here's a nice graph. Like Paul at Tetra Tech and others at Tetra Tech put this together, looking at urban centers in Baltimore, Cleveland, and San Jose, and you really see a pretty clear case of that. Biological diversity can increase within the urban gradient, but then it just falls off as it becomes more urbanized, and is really basically a cap on your potential with urbanization. So the threshold I mentioned before should be high, and I mean that there are various things you should keep in mind if you're going to do stream restoration and biological response or uplift is your goal. Be a little humble here, because do we really know what's limiting? Really spend some time trying to figure that out. We call it an urban syndrome for a reason, because sometimes we just don't quite know what factors are in play. There's the ghost of land use past. There's things that have happened in the past that are not readily apparent. So doing some investigation to that to see what effects might be going on there. And there's always unexpected consequences. Keep that in mind. This is not, again, a graph to read, but this is the Chesapeake Bay expert panel related to crediting for stream restoration, which is one reason stream restorations are very common in the Chesapeake Bay watershed, because there's a lot of credit for pollution reduction. But at the same time, when you're doing that pollution reduction, you can cause a variety of different things like depleted DO and iron block and so forth listed there on the right. So always keep those in mind. And so what should the rules be? I'm getting toward the end here. And the rules should be, in my personal opinion, avoid restoration where there's sensitive species and communities. We don't know enough to know what they need. So if they're there, we're lucky to have them and keep them the way they are. Don't do a pollution reduction where there's sensitive species or rare communities. Use the least invasive approach first. That's always a good a rule of thumb. Don't assume that erosion needs to be fixed in every situation. Lots of times people will go out and they'll look at a stream and they'll see, oh, that's pretty eroded, I better fix that. Some of the highest quality, most diverse streams in Maryland have a substantial erosion. So, maybe they would be better without that erosion, but you really need to be careful about the unintended consequences. And don't assume that all streams should look alike. Again, there's a big movement here for a lot of beaver analog systems, stream wetland complexes that we looked at with the RSCs. And while those are appropriate in many cases and can have some great value, the whole landscape shouldn't look the same. We need biodiversity requires, and historically we've had many stream types. So that should be the goal of restoration. And again, the impetus for this discussion, this synthesis, was the fact that the Chesapeake Bay program has the goal of increasing stream quality in 10% of their streams across the whole Bay watershed over the next 10 years. And so in order to do that, this is what I recommended to them at the STAC workshop. Find streams with few limiting stressors, but you know what they are and you can identify them. Mostly look outside urban settings because the potential is highly limited there. Look to fill in the graded gaps in good landscapes. So where you've got species that are there that can recolonize, you've got lesser controlling factors, thus good candidates for restoration. Remove physical barriers, add missing or diverse habitats, and maybe give it some time, give it a decade before you expect to see any improvement. And with that, I'm finished. I think I'm sort of the time frame you wanted. Happy to answer any questions.
Thank you, Mark. Does anyone have any questions for him?
Hold on, I'm on mute. Oh, hi, Mark.
No, you weren't.
I'm sorry.
That's okay, Josh.
Mark, this is Josh Gilman. I like your work and I like your matter-of-fact presentation. It makes me scratch my head a lot. I like this cartoon at the end. All of these different animals racing for the finish line. It makes me think of like agencies are thinking of stewardship in one way, and developers and DOTs are thinking of stewardship in another way. Practitioners, and I think you bring a real truth talk, science-based truth talk to the whole situation. I agree. It feels like we're trying to hurry up and measure success when it does take time. And I'm a huge proponent of avoidance and minimization, and emphasize that as a practice. I think it would be a good practice to implement. We can't feel assured that our agencies are going to find equitable offsets to losses. So I feel strongly that preservation and protection of less disturbed lands is probably a good avenue. But yeah, I just want to say thank you. This is very thoughtful and it's thought-provoking. So I just wanted to say thanks and comment. Thank you.
I really appreciate that. And it is quite a debate now, I'm sure probably across the country, but especially here in the Chesapeake, because there's been so much credit given and there are needs to reduce pollution. And because it's the best bang for the dollar or best credit for the, not necessarily bang, best credit, pollution reduction credit for the dollar is happening all over the place. So there's quite a bit of pushback now. And a lot of it is based on, are you making the biology better? And so we really are beginning to have some very frank discussions here. There's a new a law passed in Maryland to say, look at the permitting process and see how that works. And I think we're going to come out with some what we're trying to do is come out with some thresholds for intervention and some better ways to quantify trade-offs, because you're getting that. So yeah, it's I just hopefully this sort of lays it out there so that people can understand, the whole context of it, because a lot of people are just zeroing in like a laser on the pollution reduction and not thinking about the context of the whole ecosystem and how the biology can be affected.
Very good. Anyone else?
Well, as a follow-up, I'm curious, Mark, what your thoughts are on the expert panel method. It's gained a lot of attention nationally. Some of the some of it's pretty broad and incorporates a lot.
I mean, I think the expert panel approach is a good one. I mean, some people would say that the fact that there are practitioners on the panel is inappropriate, and I'm not going to make a decision or make a judgment on that. But what I would say is that, really it's goal-based. And what's interesting is the Chesapeake Bay, program, which has been around now 40 years or whatever, been doing a lot of good things, came up with this diet, which said that reducing nutrients and sediments were the key to improving the bay's living resources. And so they focused on that. And so the expert panels are focusing on what that can happen. But the bay also has, as I just mentioned, other goals, increasing stream health by 10%, increasing, fish habitat, riparian plantings, various other things out there that are also goals, which don't have expert panels on them yet. And maybe they should have them, have some, so that basically you can go out after that. And so once you begin to do that, have each of your goals as part of the crediting process, then you begin to see how they sort of meld together. Because right now it's sort of unbalanced. And while pollution reduction is really important, it's unbalanced because it doesn't look at it's always supposed to, it says, well, you shouldn't degrade the biology, you should make it better, but it's not quantified at this point. So we really need some quantification methods. Maryland Department of the Environment and DNR are looking at some more effective quantification of values, key wildlife habitats that occur along the riparian area. But still, I think no one's really got a good handle even on what things are going on inside the stream as far as uplift and whether that needs to be required. And certainly if it's, mitigate, we don't do a lot of mitigation here. And so that's why it's sort of falling this way. If it was mitigation, I think people would focus more on the uplift and making sure you've got it there. And a lot of arguments can be made one way or the other. But we're so focused on pollution reduction that we're missing the other side of the equation, I think.
Very good. I'm curious. The Oklahoma Conservation Commission works with EPA. We have a volunteer group called Blue Thumb that throughout the state of Oklahoma that go out and monitor different parameters in Oklahoma streams throughout the state, including a couple of times a year macroinvertebrate collections, and of course DO and other parameters. How many other states have programs like that?
Well, there's certainly a lot here in the East, and I assume that there are across the country. So we have something called the Chesapeake Monitoring Cooperative, and it's the Bay program initiated, and it's got a couple of different groups, and they really work with volunteers to get data that's useful. I mean, we always recognize that getting people out there and collecting and looking at critters is always good, whether the data is useful or not. We're trying to make it more useful to fill in some gaps. I mean, programs like the MDSS can go out and do 5,000 sites, and Maryland's a small state, so that's pretty effective. But large states, it's a much tougher deal to get enough professionals out there to get a decent coverage. And if we're going to do the things we say, which is avoid the high quality places and go to the gaps and stuff like that, we really need to know what the conditions are throughout the state. So I don't know much about, outside of the Chesapeake, but it's a major thing. I actually, one of my nonprofit activities is president of the board of the Patapsco Heritage Greenway, which is a nice group focused on the Patapsco Valley, which is the major river that runs down into Baltimore. And we have our own volunteer program. And because the cooperative exists, I could just tell my staff person, just go ask them how to do it. So I think that's a great resource for folks, is to talk to the cooperative. Whether or not you're in the Chesapeake, you could probably get, what are the most proven ways that volunteers can do this. So hopefully it'll continue to be done throughout the country so that we can, build on this base of knowledge that we'll need in order to make the right decisions about where to site stream restoration.
Yeah, I know they have a lot of trainings for our volunteers in the state of Oklahoma, and try to keep it very scientific and very relatable data that can be used. And so when you're looking at ecological uplift, one of the resources that someone might consider is, if you do have a volunteer organization, where are their collection sites and how long of a history do we have on those collection sites that might be near where you're doing work? That might be very, very useful.
Yeah, I would say, and we've had that debate. We have something called Stream Watchers, and Maryland DNR runs, where volunteers can go anywhere they want, and it's been probably 30,000, 40,000 tons a year. But we actually use the collections are sent to a lab, so that we do have exactly comparable stuff. So that's a little more expensive and a little harder to do if you're going to actually do those kinds of assessments. But even on a less rigorous program, if you can find the fact that there's some really nice places, we can avoid those. And I think that should be a rule. And this is one of the things I don't think we quite have yet, but the rule should be that, before you go and do a stream restoration, you really need a pretty rigorous assessment, 2 years of, stuff going on in the stream, so that what your unintended consequences could be. And if it turns out you've got a special community there, you really need to rethink what you're going to do, because you got to be careful. So, and I don't think that happens yet. I think that's one of the big easiest steps, is just to require that in every case.
Anyone else have questions or comments? Okay, well, as you can see, I'm not David. Today I'm identifying as David, but I am not him today. And unless Josh or Matt, of someone who was designated to give an inspirational talk, I think we're finished, aren't we?
Yeah, we'll go ahead and pass. Mark, thanks again. That was really it's really refreshing just to see it. You provide in a really clear perspective, and it's good food for thought. We all need to be focused on these things as we get tangled up in our weeks with our occupations. So I appreciate it, and keep doing good stuff.
Right. Well, thank you again for the opportunity. Really enjoyed it, and we'll keep in touch.
Yeah, I hope to see you
Thank you,
around.
Mark.
Yep. Bye-bye.
Okay, bye.